In mutation theory, mutations are often considered to be random with respect to fitness. And so in this presentation, we're going to kind of challenge that notion a bit and see if there's a more precise notion that we can come up with as to what is the nature of mutations. And so, as you can tell from the title, there's a little hint of what's coming, possibly external selection, and we'll get into that a little bit more as we go on. But basically, this is the question of, are mutations copying errors, or is there something deeper that's going on in mutations? So, this presentation is based on a paper recently published of the same name, Random with Respect to Fitness or External Selection, an important but often overlooked distinction. So, let's start out by looking at the modern synthesis and what was their approach. So, the modern synthesis viewed that mutations were random with respect to fitness. So what does that mean? It means that they're occurring haphazardly. In other words, they're copying errors. There's nothing intrinsically directed about where a mutation is occurring. So basically, there's no relationship between the likelihood of a mutation and its fitness effects. Now, there are biochemical biases to mutations. That is, there's nothing in the modern synthesis that says that mutations have to occur in a uniform random distribution. That's they are perfectly fine considering um, a non-random in the sense of, some bases are more likely to be mutated, some areas of the chromosome are more likely to be mutated, but because they're viewed as copying errors, there's no intrinsic association between the biochemical biases and whether or not they are more or less likely to be in favor of fitness. So again, because they're viewing these as errors in copying, and not something that the organism is doing, then there's no expectation for them to be related to fitness in any way. So if you look at Stolfus has a interesting book about mutations and randomness, and he has an entire appendix dedicated to going through the literature of the modern synthesis and showing that this is exactly what has been uh, taught in biology for the uh, since the start of the modern synthesis. Because some people, there's a lot of confusion on this because some people say, well, it's not random, but what they really mean is that it is random with respect to fitness, but there's biochemical non-randomness, or they might say that, well, there's non-randomness that occurs after the mutation. And so just being really clear about where the randomness, what does it mean to be random with respect to fitness, and what is the standard biological assumptions of what mutation is like. So anyway, so the gist of it all is that there's nothing that points mutations in any sort of organism-centric direction. There might be things that point mutations in some sort of biochemical direction, but there's nothing uh, that's kind of it's not there for the organism. It just happens to be a happenstance occurrence. So again, this is currently the dominant view even in the Evo Devo community. And some people, one of my viewers actually had objected and said that the Evo Devo group has a different view, but I went and I showed that from the 90s to the present, the standard view of how mutations themselves occur, even in the Evo Devo community, is still the same as the modern synthesis. It is that mutations are random with respect to fitness. Now, so what's the evidence that people use for randomness with respect to fitness? Well, so the evidence that people usually point to when they try to describe why they think mutations are random with respect to fitness is, first of all, mutations occur in genes that are not under selection. So you have mutations, perhaps an organism is facing a selective pressure, and it gets a mutation that's not in the gene that it's looking for. Well, or not in the gene that's going to be helpful to solve that problem. Okay. Then you also have, in many observed cases, selection pressures do not affect mutation rates. So in the Luria-Delbruck experiment, they basically showed two things. Basically, that mutations were happening at the same rate whether or not selection was present, and second of all, that selection was not selection pressure was not inducing the mutations. And so those two experiments showed that mutations were not more likely to occur when it was facing selection pressure. And then the other one is that there's a lot more deleterious or neutral mutations than beneficial ones. Uh, Genuinely beneficial mutations are very rare. We're much more likely for any given mutational event for it to be deleterious or neutral. So you might say, okay, so what's the problem? This seems like an open and shut case. Well, the problem is that the claim is actually not fully justified by the evidence, because fitness actually has multiple components, and we can simplify things by dividing fitness into two basic categories. So one is external selection. So external selection is how well does the organism fit in the environment? What is its fitness to the environment? Does it eat the things the environment provides? Is what's its relationship with the predators around? That sort of thing. How well is the organism fit to the environment? But there's a second part of selection that oftentimes gets overlooked, especially just in general discussions, and that is internal selection. That's how well an organism's parts work together to achieve homeostasis. So basically, it's saying, if you've got these internally functioning organisms, and if they don't work together, the organism dies, or at least is very sick. And so this internal selection, that the organism maintains this internal balance, uh, that if this internal balance goes out of whack, that is selective. That's a selective pressure. Um, and so and the thing that's interesting is the evidence that we get for mutation being random with respect to fitness actually only applies to external selection and not internal selection pressures. So, and the and basically what it is that these experiments, they don't have the way to take into account alternate potential genotypes that organisms don't explore naturally. So if an organism is biasing towards a certain type of mutation or away from a different type of mutation, so such that certain mutations just don't occur. Well, we wouldn't know it from these types of experiments. We need different types of experiments in order to detect that. So let me give you a thought experiment to kind of illustrate this. Of course, it's a thought experiment, so real biology is obviously different. But let's assume we have an organism that has a single gene. Okay, so this single gene can only have 7 possible alternatives, and we'll call them A, B, C, D, E, F, and G. Okay, so literally the only possible mutations are between these 7 configurations, and there's only one gene. And let's say that there's 3 available environments, X, Y, and Z. Okay, so environment X, A performs best. Environment Y, B performs best. And environment Z, you have C performing best. Now, configuration D is detrimental in all environments because it has low internal fitness. So it's not lethal, but it's going to cause problems pretty much no matter what environment you put it in. E is lethal in all environments, but not because there's an intrinsic problem, but just because it's not fit to any environment. So perhaps E is lethal because uh, under configuration E, the environment doesn't provide the right food or something like that, or E is always going to be preyed upon, something like that. So then let's say F is lethal in all environments because of problems of internal fitness. So in F, um, if you F, there's something about the internal mechanics of the organism that doesn't work under F. And then G is lethal in all environments. It's a different configuration, but it's lethal in all environments for the same reason, because of internal fitness. So let's think about this. So if we talk about mutations being random with respect to fitness, um, that means that the fitness effects don't have any impact on which um, which mutation you're going to go. So um, now, to make it simpler, let's consider the uniform random case, although it works even outside of that, but that'll make our thought experiment simpler. So uniform random case says that we're going to have 1 in 7, if we have a mutation, it's going to be 1 in 7 chance of which one, actually technically 1 in 6 because you're at a particular one, 1 in 6 chance of which one you're going to mutate to. Okay. So now if something was not random with respect to external selection, then in environment X, A would be more likely to occur because it's responding to environmental selective pressures. But what our Liederberg and Luria-Delbruck experiments show that whether we're environment X, Y, or Z, we're getting the same mutation rates for all of these. But let's say that in our studies we find out that configuration F, even though it's a valid genetic configuration, um, it's not being hit. So there's something that's preventing F from being from the organism mutating to F. Okay. Well, in that case, that increases. It means that it increases the probability that the effect of a mutation will be fit because we're no longer considering F, and so a a larger percentage of the mutations are now beneficial than they would have been. Now, that doesn't mean that an organism is more likely to get a beneficial mutation than not. It just means it's more likely to get a beneficial mutation than it would otherwise. Or another way to state that a little bit more generically is that the expected value of the fitness resulting from a mutation is higher uh, with this uh, with this mutation strategy than it would have been otherwise. Okay, um, so that's kind of the way to think about it is that the organism doesn't necessarily know what environments it's in, but it might know something about its own internal biology that tells it what the likely beneficial places to mutate are, or what the likely bad places not to mutate are. So anyway, so that's a kind of a thought experiment that kind of helps you visualize and internalize what's going on. So the basic idea behind the paper is that random with respect to fitness should be recast as random with respect to external selection. And this actually has a lot of implications on how we do biology. It is a small change, but it actually does have some longer reaching effects than you might imagine. So kind of affects our study of biological evolution conceptually, mathematically, and inferentially. And we'll look at those uh, each in turn. And additionally, so far we've just talked about how these conceptions are different. What we've established so far is that there is a difference in conception between mutations being random with respect to fitness versus being random with respect to external selection. And we've shown that the most that the experiments can do is show that a mutation is random with respect to external selection. What we have not shown yet is that mutations are not random with respect to internal selection. So we've shown that the experiments only show one of those, but we have not shown that there's reason to disbelieve the other one. So we'll talk about those right now. So some confirmed examples of mutations which are non-random with respect to fitness, even though they likely are random with respect to external selection. It's been noted since the 90s, at least, that there's a higher mutation rate in contingency genes, which are genes that kind of help you interface with the outside world, versus housekeeping genes, which are the ones that are used to maintain homeostasis. And so this is, and there's a general biological belief that the housekeeping genes are more likely to cause problems if you mutate them. So that's one area. Another one is we've also found that there's kind of a similar lethal avoidance happening in Arabidopsis and this was a paper that was out like about a year ago, and there's a distinct non-random distribution of where mutations occur, and they are not occurring in places that would cause problems. Um, so then Neisseria is an interesting one. Neisseria has an outer coat protein um, that um, that it actually has multiple alternate pseudogenes for. And basically, what happens is that the mutations for the outer coat protein occur by one of the alternate pseudogenes copying in um, its DNA sequence into the expressed reading frame in the expressed gene. And so basically, what it's doing is it's cycling through basically pre-configured options. So it's got basically a preset set of possibilities. It cycles through them, um, and that is the primary source of variation. So they're cyclical. We're not having, if you think about mutations, you usually don't think of as cyclical because the chance of returning to a previous configuration is actually pretty small. But in this case, since you have a fixed number of alternates and they simply cycle between the two, not two, there's about, I think, if I remember correctly, there are about 12 of them, maybe 20, but they cycle through them, and so the chances of returning to one is pretty high, and so the mutations behave more cyclically. So these are, and so since each of these are in fact an outer coat protein, they are, this is non-random with respect to biological function. It might be random with respect to external selection, but is non-random with respect to internal fitness because they need outer coat proteins. Again, so there's no evidence that these are selection dependent, but they are not random with respect to fitness. Now, there's also evidence coming up that there are in fact mutations that are non-random with respect to um, external selection as well. But my point here is that even if you take the ones that are random with respect to external selection, there's one, the ones that we can, as far as we can tell, they're random with respect to external selection. That does not necessarily mean that they're random with respect to fitness as a whole. Um, and so, what are the conceptual implications of this? Well, first of all, it's a mistake to view mutations as copying errors, at least in the general case. Now, I'm not saying that there aren't copying errors. Obviously, there probably are mutations that occur uh, that are, you know, just happenstance and haphazard, whatnot. Hearts have heart attacks. Every biological process has a way that it can go wrong. But the idea is that we shouldn't view mutations in general as copying errors. Uh, in general, mutation may just be an ordinary biological process. And it may just be that the reason why we thought that it was haphazard was because the mutations that we could detect when we didn't have high precision equipment, it was much more easy to detect mutations that were causing problems than mutations that weren't. Um, so along those lines, it may be that the causes behind many mutations are actually a lot more biologically oriented than previously assumed. And if that's the case, we should be looking for mechanisms. What are the mechanisms of mutations? How does the cell, what are the, how is the cell programmed to react, or how is the cell programmed to find the mutations that are more likely to be beneficial? So additionally, this affects our long-term view of evolution because our, the since mutation is the substrate for change in evolution, it used to be thought that the substrate was basically um, arbitrarily variable. If mutations are random, then they could go anywhere. But here we're seeing that mutations actually, the cell is governing what the mutation range is looking like. And so if the organisms themselves are restricting the mutation range, then that means that evolution itself has some sort of inherent internal ordering. So that's not a view of evolution that is oftentimes thought about a lot. So then there's mathematical implications to this. And so basically, as we understand these mutational modes, mechanisms, strategies, we can actually make models of them and help us understand their effects on fitness. So, for example, if you take a strict lethal avoidance strategy, you can kind of use that to model what the abstract effect on fitness is. And I go through the paper how to derive this equation. It's pretty straightforward, but I'm not going to do it here because it's harder to do in a presentation format. But the details are present in the paper. And so we, and my point here is not that this is the model or anything like that, but my point is that um, having this idea allows us to make more interesting and robust models for what is actually happening and how it relates to the fitness of the organism. And that the details of the DNA and biochemical mechanisms matter to our modeling of how mutations affect population dynamics. So again, this is meant to give a kind of be a demonstration of how you could do that. So there's also inferential implications to this. Basically, what's happened a lot in biology is people have used, even though obviously, um, even the ones who, even people who believe that mutations are random with respect to fitness, don't believe that they're a uniform random distribution. Nonetheless, you can, if they are in fact random with respect to fitness, you can use a uniform random distribution to generate a proxy for what's actually happening in biology. And the reason for that is that the uniform random distribution will basically give you an expected value for fitness. Um, and so uh, so even though there might be biochemical biases, uh, using you don't have to know those ahead of time. You can use a uniform random distribution to get you an expected value for the mutational range, and then you can use that to predict fitness effects and other things. And so basically, what was happening is that in many areas of biology, uh, randomness of some form or another is used as a proxy for what is occurring in cells for mutations. But if that's not actually the case, then we actually have to rethink some other aspects of biology. So um, the one field that does this a little bit more heavily than others is the study of the distribution of fitness effects. And so what happens a lot here is that they'll mutate proteins at random and figure out what's the distribution of beneficial and deleterious effects of mutating these proteins at random, and use that as a proxy for the distribution of fitness effects of mutations in general. And the fact is that if mutations are not random with respect to fitness, then this procedure does not work. Or if it does work, it's only incidentally working. So anyway, so final takeaways, mutations, as far as we can tell, they're not random with respect to fitness, but many of them do seem to be random with respect to external selection. And those are distinct concepts, and it's important to get that distinction correct. And so while this is somewhat of a small change in perspective, it actually has further reaching effects on how we view biology and evolution than you might otherwise anticipate. Um, but additionally, this new perspective can help us better model mutational processes. It kind of gives us a starting point for understanding, and once we understand better how the mutational process works, we can model it better, and we can model its effect on fitness. And then finally, we need to look at various areas of biology that have been influenced by the idea of mutations being random, and we need to revamp them. And so, whether it was relying on this idea implicitly or explicitly, we need to look and see which parts of biology need to be fixed up and replaced by better models. So, thank you for listening, and I hope you enjoyed it.